Good evening,
Am I on?
ma'am. Ma'am, your voice is breaking.
Okay. Am I audible now?
Yes, ma'am.
Okay. So, good evening, everyone. It's an honor to welcome you all to today's workshop on Truth in the Teeth, Species Identification in Forensic Science, organized by Shivi Forensics. I am Vaishnavi Pawar, student of Forensic Science at MGM University and the coordinator for today's event. It's truly a privilege to host this insightful session and connect with brilliant minds in the field of forensic science. We are honored to have with us Dr. Anjali Sehrawat, currently serving as a project assistant at the National Forensic Sciences University under the Defense POW Accounting Agency project. Maam holds an MFC in Forensic Odontology, a Bachelor of Dental Surgery, and a professional diploma in Forensic Archaeology, a rare blend that bridges dentistry, forensic science, and archaeology. She has also served as an assistant professor of forensic science at Devbhoomi Uttarakhand University and holds a German patent for a non-destructive elemental analysis method for species identification using teeth. Her research and presentations have earned her several national and international recognitions, including the Best Paper Award at the 20th IFO Conference and the Award of Excellence at the 4th International Forensic Science Conference. On behalf of Shivi Forensics, we warmly welcome you, ma'am.
Thank you so much.
Now, without further ado, I invite you to take over the session and share your valuable insights with us. Ma'am, the platform is all yours now.
Okay, I will just share my PPT. Just a second. Is my screen is visible now?
Yes, ma'am, it is visible.
So, good evening, everyone. Thank you for joining us our DPA project. Today, we will explore how forensic odontology can help us in distinguishing human from non-human dental and skeletal remains. It is a foundational question in many medical fields, anthropological or archaeological, and wildlife forensic and disaster victim identification contexts. So, from start, let's begin with the PPT and the topic. So the first question is whether it's human or non-human. So let's begin with a scenario as investigators recover a small fragment of tooth or a bone from a site, perhaps after a fire, mass disaster, or wildlife crime. The first question must be: is the fragment human or not? So the answer determines the entire path of investigation. It determines whether a legal death investigation is needed or whether resources are allocated differently. So today talk I will show how we use gross anatomy, microscopy, chemistry, radiography, etc. to answer that question and what their strength and limitations are. So human versus non-human, it's more than just a tooth. So this distinction has uh, real world consequences. If the human if the tooth is human, the police must treat it as a potential evidence in a crime. But if it's animal, it could mean nothing criminal at all. So in disaster victim identification, time and resources are precious, and misidentifying animal remains as human waste are valuable efforts. So, and in legal context, mistake can cause wrongful assumption. According to a study from Sydney indicates that over 70% of skeletal cases that received were ultimately non-human and yet require significant investigator time. So it is a wrongful assumption and misidentifying of human and non-human remain. Another example is in 1993 in US, animal bones were misidentified as human, leading to an unnecessary homicide investigation. In it was only later, with the help of forensic anthropologists, that the error was corrected. This shows why species identification is not optimal, but it is also essential. So, moving on to the overview of forensic odontology. Forensic odontology is the application of dental sciences in law. What makes it so powerful is durability of the tooth. So, according to the FDI, forensic odontology is that branch of dentistry which, in the interest of justice, deal with the proper handling, examining of the dental evidences, and with the proper evaluation and presentation of dental findings. So, but the question is why teeth? So, in forensic odontology, teeth are the hardest and the most reliable structure in the body. They withstand physical forces, chemical corrosion, biological breakdown. In fact, they can be exposed to a temperature as high as 1600 Fahrenheit without appreciable loss of microstructures. This means they can survive virtual intact long after soft tissue or skeletal bone have been destroyed by decay or even incineration. Another reason, they are invaluably and unique. So, human dentition is almost as individual as a fingerprint. Even environmental influences like fluorosis also leaves unique imprints taken together. This makes it extremely rare to find two identical dentition. So, teeth not only endure, they also preserve a lifetime record of identity and environment, making them one of the most reliable tools in forensic science. So, at what fields we can use forensic human teeth is like forensic implication of human dentition. So, first is uniqueness of dentition as forensic evidences. Beyond species identification, teeth also help in personal identification. Each individual dentition is unique due to its variation in tooth size, shape, alignment, restoration, and pathological changes like caries and fluorosis. Just as fingerprint differ not to individual dentition are exactly alike. This uniqueness allows forensic odontologists to match dental records of missing person with unidentified remains when even when the soft tissue are completely lost. So next is sex and age determination. So when we look at a dentition for sex determination, the size and proportion of the teeth often gives us the first clue, generally whether it's male or female. So generally male has larger teeth than female and both in mesiodistal and buccolingual dimensions. Teeth are also valuable in age estimation. The degree of teeth eruption, adhesion, secondary dentition, decomposition, and root transparency all provide a clue about the individual chronological age. In subadults, tooth development stages offer high precision, while in adults, regressive changes are also used for age estimation. Now, the next is ancestry. Dental traits can indicate ancestry. Uh, Asian often resemble shovel-shaped incisor, shortened root, extra molar, and more enamel pearls on premolars. Whereas European frequently show cusp of carabli on upper molar, and African may have fourth molar. Some groups show cultural modification like chipping or filling, and rare traits like male in Buddha's tribe never developed teeth are also noted. So, bite marks analysis examine the impression left when the teeth contacts a surface. Individualization factor include trauma or a missing teeth or a dental disease like caries, artesian, or root absorption, as well as anomalies such as micro or macrodontia, hypodontia. crowding, impacted teeth, and enamel hyperplasia. In forensic cases, bite mark can also help in distinguishing animal bites by analyzing the dentition pattern in the dental formula. It will be possible to identify species responsible for the marks. So, moving on to the comparative dental anatomy. This slide introduces the foundation of comparative dental anatomy and how tooth forms have evolved across species. Comparative dental anatomy is the key for identifying whether a fragment belongs to a human or an animal. For a single cone shaped teeth in early vertebrates to complex occluding molars in mammals. Each stage represents a step towards greater efficiency in food processing. Human and other arthropods ape show the most advanced pattern like quadritubular molars, which is highly diagnostic in forensic identification. So, among living species like anthropod apes, chimpanzee, gorilla, and orangutans are the closest human through their large canines and diastema differentiate them. So, recognizing these patterns provide context when analyzing a fragmentary remain. So, this is the image where you can differentiate the different type of dentition. Is my cursor is visible?
Yes, ma'am.
Okay. So this is the dentition of an omnivorous, that is mammal. So it has teeth based on different that herbivorous. Or in carnivorous, we can see that the sharp canines are present. So based on that, we can differentiate and appreciate the anatomy in different species. So moving on to the haplodonts. Haplodonts class represent the most primitive tooth form. It's a single conical cusp. These are typically seen in fish and amphibians. So, like crocodile or alligator, you can appreciate these type of dentition. The jaw open and close like a hinge, so the teeth never truly occludes. Their main role isn't chewing, but grasping or tearing, often for prey, capture, or defense. So this kind of dentition is present. It's kind of conical teeth present in haplodonts. So moving on to the trichodonts. Trichodonts class evolution add complexity. So three cusps aligned in a row. It is aligned in a straight line kind of structure in which the central cusp is dominant with smaller cusp, one before and one behind it. So the main cusp, that is central cusp, is longer. I will use it. Wait a second. So in trichodon, the dentition is like this. In a straight row having three cusps. So the main cusp will be large and with two small cusps. This form improves shearing ability, allowing better food processing than simple grasping. So pure trichodon dentition are rare today, but element of this pattern is mostly seen in some carnivores such as dog, where the posterior teeth are adapted in for slicing meat. So in some dogs we can appreciate this pattern, but due to evolution it has been extant. So the next type of dentition is tritrabecular class, and that is the tritrabecular class make a critical evolutionary advances. Here, the three cusp forms a triangular rather than a straight line. It's similar to the trichodont class, but it's in a triangular form. So the three cusps are arranged in a triangular form. So this arrangement allows partial occlusion, meaning the teeth of the upper and lower jaw start to make some functional contacts. With this, animal could not only shear but also grind their food. It's characteristic of early mammals when we were in our Homo sapien context. It was present on that time, and you can see the traces of this structure in certain primates also, like ape and orangutans. You can appreciate this kind of structure. The quadritubercular class is where the full occlusion and functional articulation develops. So the molar now have four cusps arranged in a rectangular or rhomboid pattern. This allows for the complete contact between upper and lower teeth chewing. Human and apes have this kind of pattern, enabling for grinding, crushing motion, and that defines us as a omnivorous. So this kind of, if we see our molar or the posterior teeth, we have four cusps present on the molars. So that kind of structure is called quadritubercular class. Okay. So moving on to the pattern of dentition and the tooth attachment. Species differs not only in the shape but also how their teeth are replaced and attached. So based on the type of eruption, polyphyodont species like fish and reptile continuously replace their teeth. Like think of shark, which can lose or grow 100 teeth over a lifetime. So if their one teeth exfoliates, the other teeth will erupt, and that cycle continues for the lifetime. So human are diffodonts, that meaning we get only two set of teeth, that is milk and adult teeth. And some mammals such as certain whales are monophyodonts, which have one generation of teeth, that if their teeth exfoliates, they will not get another set of teeth for the lifetime. In the terms of form, reptile and amphibians are usually homodonts, where every tooth like the same structure, while the mammal are heterodonts, the incisors and canine premolar and molars, each serving specific role for the cutting, tearing, and grinding. So, and based on the attachment also tells us a lot. So in acrodont species like snake, teeth sits just right on the bone surface, so there is no specific attachment in snakes or acrodonts. In pleurodont, such as lizard, the teeth are attached along the inner side of the jaw, but it's not completely attached, so that is pleurodonts. And in human and mammals, thecodonts, their teeth are securely anchored in socket. So our teeth are securely anchored in socket and giving the most stable kind of attachment. Moving moving on to the why this matters in forensic science. So just a second. So, identifying isolated or fragmentary bone can be extremely difficult in archaeological or forensic contexts. So, numerous osseous materials such as wood, pottery, plastic, or even stone can sometimes be mistaken. Sometimes be mistaken as stone, or sometimes be mistaken as fragments of human or non-human bone. So this analysis, we take it into three steps, that is gross skeletal analysis, microstructure and macrostructure of the bone. So, moving quick to the dental anatomy, dental formula provides a quick way to identify species based on the number and the arrangement of the teeth. So, human have 32 teeth with a dental formula of 2123, whereas apes share the same dental formula, that is 2123, but typically have larger canine and diastema, that is space between the teeth. The So that differentiate apes from human. Even dog have 42 teeth with a dental formula of 3142 in the upper jaw and 3143 in the lower jaw. Cat have 30 teeth with dental formula of 3131 and 3121. Cow lack upper incisors and instead have a dental pad. Their formula is 0033 3133, and pig have 44 teeth with bonodont molars that with a dental formula of 3143. Bonodonts are the kind of molars which are posterior teeth with less sharp cusp tips. So their cusps are not that much sharp. It is used for grinding only or chewing food. So comparative jaw and tooth morphology across the species. Human have a parabolic dental arches with thick enamel for grinding, ideal for an omnivorous diet, whereas dog and cat have a V-shaped and a circular arches respectively with the sharp teeth for slicing and puncturing meat. Their carnassial teeth act like scissors. So cow possess a broad flat jaw with a ridged molar for grinding cellulose-rich food or leafy food. Not lack, note that they lack upper incisor, and instead of that they have dental pads. So pig are interestingly have bunodonts molar resembling human molar, making forensic identification tricky when only molars are found. So ape have U-shaped jaw with large canine and diastema, a space that accommodates the opposing canine used for feeding and social display also. So morphological methods, it is my study which I have done for the human and non-human identification. So morphological analysis represent the foundational approach in forensic dental species identification. So the qualitative assessment examine the visible structural characteristic that distinguish human teeth from those of other species. So this method relies on careful visual observation and anatomical knowledge to identify key diagnostic features. So the features could be tooth shape and size. So human have smaller scapulated incisors with low rounded cusps adapted for omnivorous diet, whereas carnivores have pointed and sharp shearing cusps of tearing meat. And herbivores have a broad molars with complex ridge for grinding plant materials. Whereas the crown root ratio, human typically display a crown to root ratio of approximately that is 1:2, providing stability for mastigation. But often animal have a longer root relative to crown height that can be 1:3 or 1:4 in the ratio, providing enhanced traction for specialized dietary functions. So you can appreciate it in the picture, like the dentition of human and animals. It's the dentition of ape. This is a dentition of ape. So how different it is? The size of the tooth is quite different than animal, but the number of teeth and are quite similar. So it has two incisors, one canine, two premolar, and three molars. Similarly, human also have two incisors, one canine, two premolar, and three molars. So dental formula is same for ape and human, but their size and shape is different. So enamel thickness. Human enamel is notably thick, providing protection against various diet and extensively chewing. Animal enamel tends to be thinner, with more frequent dentine exposure reflecting different masticatory demand. So human enamel would be thicker in comparison to non-human. Root configuration. Human exhibit multi-rooted molars with distinct bifurcation, while animal species represent single-rooted molar adapted to specific functional demands. So we have single-rooted molar in comparison to non-humans. So research finding. In control studied, crown contour and root bifurcation pattern reliably distinguish human molars from canine species. That is, dogs. Dog teeth consistently demonstrate elongated conical roots with sharper cusp morphology. So this is my sample study for the morphological method and qualitative assessment. I have done the quantitative assessment also. So we have gone for the visual examination of different kind of species, including dog, cat, cow, pig, buffalo, goat, and sheep also. And even we have taken the dental impression so that we can go for the visual examination later on stages, so that if we have missed any dental feature or the anatomical feature during our visual examination, as the species might react, don't cooperate at that time, so we can use that dental impression for later studies also. So moving on to the morphometric studies, uh, morphometric analysis advances beyond subjective observation to provide objective, measurable data for species identification and differentiation. This quantitative approach employs standardized measurements and statistical analysis to establish reliable diagnostic criteria like digital caliper, imaging software, and three-dimensional scanning techniques enable precise documentation of dental dimensions. So, in this dental dimension, like mesiodistal width, buccolingual diameter, crown height, and root length are systematically measured using standardized protocol with vernier calipers. These measurements create a quantitative profile for unique to each species. So statistical discrimination, how we have used statistics is like discriminant functional analysis, principal component analysis, and logical regression model process multiple measurements simultaneously. We achieve classification accuracies exceeding 95% of control studies. So the buccolingual length and the mesiodistal diameter and the intercanine distance is also used for this study, and the root length of different species. The root and the crown ratio is also measured for the identification. So, digital documentation, the three-dimensional surface scanning, photogrammetry, and computer-aided design. That is, CAD software create a permanent digital record. So we record it in a digital format even after scanning. So enabling a retrospective analysis for comparative research, because it's an ethical consideration. So if we have a retrospective analysis and sample, so we don't need to provide an ethical certificate for our studies. So clinical application, that is morphometric database containing measurement from diverse population, enables us for comparison of unknown species against the established norms, facilitating rapid and accurate species determination in forensic context. So this is how we have done our intercanine or the other measurements. You can appreciate it's the sample of dog and the cat. So the difference between the measurement difference between cat and dog, if our sample falls between this kind of range, we can easily appreciate and we can differentiate which species it is. So it is very easy for us to differentiate between different kind of species if we have a large database for quantification assessment. So moving on to the skeletal anatomy of human versus non-human. I'm sorry. So the skull tells the story. So brain to body ratio. If human possess an extraordinary large brain compared to body size, resulting in large bulbous cranial vault, that dominate the skull appearance. So, if we compare, we have a large cranial size in comparison to non-human. Non-human has small brain, so their cranial size is also small. Based on that only, our facial projection is also dependent. So, human faces are remarkably small relative to our cranial walls. So, if we see our faces, so in comparison to our cranium, our face is kind of small in comparison to our cranium. And in non-human animals show the opposite pattern with large faces and small vault. So their vault is quite small, but their face is quite large. So even the muscle attachments, human vault musculature is less developed, and animal often display prominent sagittal and occipital crest for powerful jaw muscle attachment. Moving on to the vault bone structure. So the large human brain creates more curved vault bone with distinct layering of cortex and dipole, that is spongy bone between inner and outer table. This layer is often visible in fragments and in much more pronounced than in non-human animals. And the orientation and opening. So in human crania are oriented in a vertical axis. Our cranium is oriented in a vertical axis with orbit position in front. So our orbits are present in front above the nasal aperture. This is the nasal aperture. And the foramen magnum, that is where the our spinal cord enters, sits inferiorly at the skull base. In non-human, the crania follow a horizontal axis with orbit located laterally and behind the nasal aperture. So our orbits, mostly in animals, orbit are placed on the lateral surface, and the foramen magnum is present posteriorly, that is behind, reflecting the quadrupedal locomotion. So it is like how our anatomy is. But how will you identify when you get a skull or a bone or a cranium? So this is how you can identify. So read between the clues. It's like curvature. The human vault bone display greater curvature due to the bulbous cranial shape, while the animal vault bones are flatter and more angular. So human vault is more curved than the non-human. So the thickness pattern, the three-layer structure, outer cortex, dipole, and the inner cortex is much more distinct in human bone in comparison to non-human. So you can appreciate it in this picture also. And the inferior surface. Look for the relatively smoother inferior surface with possible venous grooves. In human, we have a smooth interior surface in comparison to non-humans versus the complex surface morphology of non-human. So if we see the human interior surface, we can see the venous grooves present on the internal structure of the vault. So all the veins and the arteries passing through the vault and the brain arteries, we can appreciate it on the structure of the vault. But in comparison to non-human, it is not that much smooth, and the structure is not that much appreciated. So overall density also differentiate between human and non-human. So human vault bone have thicker dipole relative to cortical bones compared to the thinner, more compact structure of animal vaults. So we have more thicker dipoles in comparison to non-human. Moving on to the postcranial skeletal differences. So the way human and most non-human animal moves, bipedal versus quadrupedal, meaning on two legs or four legs, lead to the significant differences in the postcranial skeleton. Recognizing these differences in crucial when identifying isolated bone in forensic and archaeological contexts. So human adaptation is singular vertex Like human walks upright on two legs to support this skeletal has several adaptation. Like the cranium is centrally placed over the spine to balance the head, and the spine has four curves to absorb the distributed weight. So the pelvis is broad and short to stabilize the trunk, that is the upper portion of the human, and the femora angle, that is femur, is angulated inwards towards the knee. So the proximal tibia are thickened to bear the weight, while the feet have the dual arches for the shock absorption, and the upper limbs are glacial, not weight bearing, but have a greater range for the motion for the manipulation. So our lower limbs have the weight bearing bones, and the upper limbs are for the manipulation or the motion. Whereas in non-human, that is quadrupedal feature, most non-human animal walk on all four limbs. Their skeleton are adapted for stability and locomotion across the dual axis. They usually have no spine curvature, are robust, and often have fused radius and ulna and tibia fibula bones for strength. So as if we see in human bones, we have separated radius and ulna. It's not fused, but in non-human, the radius and ulna are fused bone for the strength. The scapula are oriented along the axial plane for to facilitate the quadrupedal movement. So if we see in the image, it's the image of uh, deer representing its narrow and long pelvis, and in human it's broad and it's shorter. So we can say human have a bowl-shaped pelvis in comparison to non-human. Non-human will have a squarish kind of a or box-shaped of pelvis as they walk on a quadrupedal motion. Moving on to the scapular orientation, human scapula are broad and oriented inferiorly, whereas in animal, scapula are long, narrow, and oriented towards the spine. So this is the scapula of non-human, and this is the scapula of human. So the outer blade is kind of wider in human, and it's narrow in non-human. So moving on to the long bone morphology, human are more glacial, animal are more shorter, broader, and more robust with the density relative to their size. So there are few special consideration for the identification and kind of its confusion while identifying. So certain skeletal remain present unique identification challenges, particularly infant human bones and avian bones, that is birds, which can easily be confused with other material or species. So in infant human bones are among the most commonly misidentified human remains. Unlikely adult bones, they are incompletely ossified. Epiphyses are often unformed or unfused, giving rough, indistinct edges. So, multiple ossification center increase the total number of bone elements, many of which cannot be assigned to a specific bone. So, they are different part of bone because they are not fused at that time. So long bone diaphyses are thin and lack the adult trabecular structure, making them appear fragile and sometimes confusing when found in isolation. Whereas in avian bone, bird bones are light-weighted with with very thin cortical walls and minimal trabecular structure at the end, and the adaptation for flight. So they are bipedal like human. They also walk on 2 feet. So, but they're fully developed epiphyses and pneumatized structure, that is for air-filled sacs structure, distinguish them from the infant. So they are differentiated due to their developed epiphyses. In non-human, we the epiphyses are fused, but in human at that age, epiphyses are not fused. In cranial vault confusion, juvenile cranial vault bones can easily be decided into two parts. So, the petrous portion of the temporal bone being easily dense tend to survive burial better than other infant bone, frequently appearing as isolated fragments that are difficult to identify without careful analysis. So, these are few bones that I have incorporated. This is the bone of turkey. So it looks similar to the humeral bone of an infant. But how we can differentiate the epiphyses in turkey bone is fused and the epiphyses are closed. But in the in the human bone, the infant bone, the epiphyses are not fully developed. So we can differentiate based on that. Similarly, in this image, this is the cranial fragment of the infant, and this is the uh, tortoise carapace. That is the upper structure of the tortoise. It's kind of similar, looks similar, as they both have that uh, they don't have that dipole bone. Infant don't have the dipole bone. Because of that, it looks kind of similar, but we need to differentiate between this, so a thorough identification need to be done. Similarly, the petrous portion of the infant temporal bone, it's dense and often the only surviving cranial element. So moving on to the bone macrostructure. We have done with the microstructure and the anatomy. So moving on to the macrostructure, that is density and the internal architecture. Beyond gross anatomical feature, the internal structure of bone provides critical diagnosis information. The macrostructure differentiate reflects fundamental evolutionary shift in the human evolution. So, evolutionary context. So, body size increase at the origin of gene Homo, but skeletal structure become lighter to balance the energy demand of the larger brain and bodies. So, gaugational process fundamentally alter the bone density and architecture. So, I will explain it how. So, the density differences in non-human animal bone have greater density. So the density of non-human bone is high. It's large, robust, and heavy in comparison to non-human bone. So the density is relatively high, uh, related to the size of non-human human bone. So they have less porous, more compact, and thicker in cross section than the non-human bone, a reflection of different biochemical demands. And the cross section cortical thickness in humeral and femoral bone. A cortical thickness is approximately one quarter of the total diameter. So it's the human bone, and this is the non-human bone. So you can appreciate the diameter. It's the cortical bone. So it's one quarter, one fourth of the total diameter. And in animal limbs bone, the cortical thickness is about one half of the total diameter. So you can appreciate it's more thicker on this side. It's kind of similar. Twice the thickness proportionally. So moving on to the medullary surface characteristic. One of the most reliable macrostructure in differentiating the human and non-human bone. The difference appear in the medullary cavity of the bone. Diaphysis, human long bones, display a web of trabeculae covering the interior medullary surface, creating a rough, spongy appearance. So, this trabecular network provides a structural support while minimizing weight. So the stark contrast, trabecular bone is largely absent from the interior of the non-human bone. So this web-like structure is present only in human and not present in the non-human bones. The medullary surface is very smooth and compact, reflecting the different biochemical requirement of the quadruped locomotion and the need for the human cranial vault bones exhibit thick dipole and relative to the cortical bone layer. This three-layer sandwich structure provides the protection for the large brain while minimizing the weakness of the vertebral column, allowing the wearable of the vertex. So, whereas the non-human vault thin, compact function with the less pronounced dipole. So, how we will examine this? So, this is the anatomy, but after this, how we can examine this is like examine the cross section. So, you have to look for a broken end and a cross section of the long bone to assess the cortical thickness relative to the diameter. As human have a inner cortical diameter, the diameter is 1/4 of the cortical thickness. It will be 1/4, and this is to the diameter in comparison to the non-human. So check for the medullary surface, inspect the interior surface of the for the presence of human or the absence of trabecular bone network in non-human bones. Then assess porosity. Human cortical bone and non-human bone appears more compact and denser. And evaluate the vault fragment. In cranial fragment, look for the thick dipole layer versus the compact structures in non-human. So moving on to the microscopic analysis, that is histological analysis. When gross anatomy and macrostructure are inclusive, microscopic examination of bone tissue provide definitive identification. So, while not only a practical in field setting, understanding histological differences is essential for forensic aspect also. So, whenever human osteon pattern, we are studying for human bone, osteon pattern in human, cortical and trabecular bone are scattered and evenly spaced throughout the tissue. This random distribution creates a relatively uniform microscopic appearance. So, if I show you, this is a kind of osteon pattern present in bone. It's in a form of a concentric circle and it's uniform throughout the bone appearance with no orientation or alignment of eversion canal. Whereas in osteon bonding of non-human osteon and that align in distinct row or bands like this organized pattern called osteon bonding. So if we see, mostly it's in the form of a plexiform pattern, plexiform bone. So like we have in human bones, it's in a concentric circle, but in this non-human bone, it's not in a concentric circle or not in a specific manner. So it is a clear indicator of a non-human origin or reflect different patterns of bone remodeling and biomechanical stresses. In plexiform bone, some non-human animals, particularly large mammals, develop plexiform bones with a rectanguloid structure. So in that kind of bone, the osteon pattern is like this. So it's in the form of bricks. You can imagine a brick kind of pattern in plexiform bones. It is also a type of pattern present in non-humans. So this specialized tissue allows for a rapid bone growth and is diagnosed for non-human origin when present. So how we will interpret this? So while osteon bonding or a plexiform bone, so definitely indicates non-human bone, the interpretation of scattered osteon distribution requires caution, and Ubleker also emphasizes that the considerable variety exists between the different species and even between the different bones of some species like non-human animals. So additionally, health state, bone remodeling history can also affect the pattern in bones, human and of human and non-human both. Juvenile bone pathological bones and heavily remodeled bone may also show a atypical pattern that is not a specific pattern that will cause different interpretation. So moving on to the microscopic examination of teeth. So we have studied the microscopic examination of the human and non-human bone. Now moving on, how teeth will differentiate and help in differentiating human and non-human bone. So the microstructure of teeth provide valuable information for distinguishing human from non-human remains. In human, the enamel, the hardest and the most mineralized, shows enamel rod arranged in arcades and keyhole pattern. So in human, the enamel rod will be present in this kind of pattern. So this is a keyhole pattern, forming a strong interlocking network. In contrast, animal enamel varies. In in dogs show linear parallel enamel rod, while pig exhibit elliptical or irregular pattern. So these patterns reflect different masticated adaptation and are often visible under scanning electron microscope or polarized microscopy. So this is the imbricated pattern of a rhinoceros, and this is the pattern present in mouse. So, if you want to do a study on the pattern of enamel rods so between the different species, it is very easy. You just need a clean tooth. You just take the tooth from any species, just clean it, and use a cellophane tape. Apply it on the teeth, remove the teeth carefully, and apply that on the slide, and you can see the enamel rod patterns easily on the microscope. So it is a very easy kind of study, but need a lot of precision, as every species has different enamel pattern. So in dentine, the dentine located beneath the enamel is a tubular mineralized tissue extending from the pulp to the enamel. In human dentition, the tubules are uniformly distributed and regularly oriented, supporting the balance, grinding, and chewing. In animals, the dentine show variable orientation and irregular densities. So depending on the dietary habit. So example, herbivores show tightly packed tubules while the carnivores show wider and irregular spacing. So this is the intertubular dentine present in human. So it is present in a sequence. Whereas in human, it varies between herbivores and carnivores based on their diet. So the cementum covering the root plays a key role in the tooth attachment. Human cementum show multiple incremental lines and secondary deposition in different stress and function over the time, whereas in animal often have thinner cementum with fewer incremental layers as their teeth experience different mechanical stresses and wear patterns. So, in pulp, the pulp is a soft connective tissue containing nerves and vessels. Uh, all in human, pulp chamber are relatively smaller, while in animals or in carnivores, the pulp chambers are large. So, as depending on the size, we can correlate it. Like human teeth are small, so the pulp chamber will also be small in comparison to non-human, as their teeth size is quite large, their pulp chamber will also be large. So moving on to the elemental and the spectroscopic study, it is very important. is also a part of my research. So I have patent on this study, that the I think so. In the starting, it is mentioned that I have a German patent. This is the study which they were talking about. So elemental and the spectroscopic analysis provide a chemical and the structural fingerprint of the tooth and bone, so helping differentiate between human and non-human remains even when the morphological or histological are inconclusive. When we are unable to differentiate from histological or morphological features or the morphometric features, so we can use the elemental analysis for the identification of species. So this method are based on the composition, the primary mineral component of enamel and the bone. So in human, the calcium and phosphorus ratio is usually close to 1 is to 6-7, reflecting the composition of pure hydroxyapatite crystals. In animal, the ratio varies due to the differences in diet, and also as the enamel thickness is less in non-human. So herbivores often show higher strontium and lower zinc level, while the carnivores tend to exhibit elevated zinc and magnesium because of protein-rich diet. So whatever we eat or whatever we have, it reflects from our teeth. So sometimes we say, na, whatever you do, it reflects from your smile. So it's similar. So the techniques such as FTIR, EDXRF, or EDX and EDS, XRD, and ICP are commonly used. By FTR, identify phosphate and carbonate bands which reflect the crystal lattice structure of hydroxyapatite. EDX determine the elemental composition. And XRD analysis, the crystal arrangement and the crystallinity index, which can differ between the species. So these are the results of my study. So we can appreciate that the human have 7.639% of the calcium and 24.32% phosphorus present in the teeth. So if we have the range between all species, and if we analyze the sample in the EDXRF, within 2 minutes we can get a result without destroying the sample. So sometimes we get a case like we get a small tooth sample and they ask us to identify the species, but it is very difficult. So as we are not able to destroy it for the DNA identification, so they always say ki do it morphologically or from some other aspect. So sometimes it's difficult to identify it morphologically, so we can go for a better method, that is elemental analysis. That is an indestructible method for species identification. It also shows that uh, in Shivi only in the silver present, none other species the silver is present. Similarly, barium is only present in buffalo. So if we find some species and we find that it is only having barium, we can say that it might be buffalo. So it will narrow down our search and identification. So it is kind of a helpful study. So this is how we process our samples. You just need to take the sample, cover it in a box with a cellophane paper so that it will create a vacuum and place it in the EDXRF machine. And within the 2 minutes, you will get your results. So you can identify which species it belongs to. So moving on to the radiological analysis, I have done studies on radiological analysis also. I have quantified the radio density in numbers, which is quite difficult, as everyone says ki we can just see it's radio dense or radio opaque, but we cannot convert it into numbers. But now we have done it. So soon a patent will also come for that. So, radiological analysis provide a non-invasive method to examine the internal structure, density, and architecture of bone, assisting in the distinguishing human from non-human remains. These methods complement morphological, histological, elemental analysis, especially when the sample are fragmentary, degraded, or incomplete. So the key techniques are X-ray imaging, CBCT or CT, and postmortem CT. So in X-ray imaging reveals the bone density, cortical thickness, tubercular pattern, and gross structural features. Useful for rapid screening and fracture detections. In CT, it offers detailed 3D reconstruction of bone microstructure, allowing precise measurement for the cortical thickness and medullary cavity and internal trabecular network. And in postmortem, the non-destructive scanning of the skeletal remain to assess the fracture and trauma or anatomical analysis without the physical alteration of the specimen. So how we will, it's key what technique we can use, but how we will compare between the human and non-human. So we need to look for the bone density and the porosity. So human bone typically have the lower density and higher porosity compared to the non-human animals, which show denser and robust bone architecture. It is similar for the tooth also. So human tooth has lower density in comparison to non-human. And we can say that bovine tooth. So that is cow or buffalo tooth has the highest bone density. So cortical thickness, non-human bones such as those from cow, pig, and dog generally have thicker cortex, whereas human exhibit thinner cortex relative to the bone diameter. And the structural variation, differences in tubercular orientation and cortical layering and medullary cavity spaces, that is also explained in the macrostructure of the bone. Additional diagnostic feature for the species identification. So I would say that uh, human non-human identification is not based on not a single method. It's based on the integrated forensic approach that include morphology, that is field observation, rapid visual assessment for gross anatomical feature enables preliminary classification, and direct subsequent analytical strategy. That is, it's the basic of the analysis, or the visual assessment is the basic of the assessment of any kind of specimen. Then morphometry, quantitative validation through standardized measurement and statistical analysis provide objective and reproducible data supporting refining and morphological impressions, whereas microscopy, like tissue level confirmation, revealing diagnostic historical architecture invisible to the macroscopic examination, particularly valuable in fragmentary or degraded specimen. So, elemental and radiological analysis, that is chemical and structural profiling through the spectroscopic technique, which provides species signature based on the elemental composition and the radiological analysis. So, in the last, I would like if someone is able to identify what type of dentition it is, as I have mentioned the trichodont, tritubercular, haplodont, and quadritubercle. So, if you are able to identify, I am quite succeed what I have taught you today. And in the end, I would like to end my presentation with a small take home message. That is, in forensic odontology, the power lies not in one test, but in the integration of many. Morphology offer rapid field assessment, microscopic and elemental studies provide definitive differentiation, and radiological and molecular tool validates finding. So, following standardized procedure and maintaining through thorough document is essential for credibility in court and research. So, most importantly, when the evidence is ambiguous, Always side for the caution and assume the material could be human until it is otherwise. That's both ethical and a scientific approach. Thank you. That's all for today.
Thank you so much, ma'am, for the wonderful presentation. It was truly enlightening to learn about the scientific approaches
Yes,
and real world applications of forensic odontology and species identification.
thank you so much.
On behalf of Shivi Forensics, we extend our heartfelt gratitude for your valuable time and efforts in joining us today and sharing your expertise. We hope to have the pleasure of interacting with you again in the future.
Hope so. It's fruitful for everyone who have attended the session. I tried to explain non-human identification. We hope we can develop more new methods as India don't have that specific database, and for non-human it's not present in the world also. So we can collab and do more such researches, and we can come forward with such more methods.
For sure, ma'am. Thank you so much, ma'am.
Thank you so much.